the polymerase chain reaction, commonly known by the acronym PSR, is a molecular biology technique that allows the multiplication of nucleic acid fragments, of which the initial and terminal nucleotide sequences are known. Amplification by PSR allows the quantity of genetic material necessary for subsequent application to be obtained in vitro very quickly. Very often, biologists need to amplify DNA, that is, to produce many copies of a specific gene.
These two ways are the technique called cloning, in which the cells are duplicated with the DNA they contain. An alternative is the PCR, the polymerase chain reaction, with which it is possible to duplicate only the nucleotide sequence in the test tube using the enzyme DNA polymerase.
Polymerase chain reaction is a circular process made up of the repetitive stage. 1. The double-stranded DNA stranded subject to heading separated by denaturation into unstained strands. 2. The primer is added to the solution together with a mixture of free nucleotides, allowing the solution to cool. 3. The DNA polymerase catalyzes the production of new complementary strains. Every cycle of the number of DNA fragments is identical to the starting strain's doubles.
To form the complementary molecule, it's necessary to separate the double helix of the DNA to allow the DNA polymerase to insert itself. To make this happen, it's required a temperature of 19 degrees, which denatures the DNA, that is, to break the hydrogen bridge. However, such a high temperature destroys the DNA polymerase that cannot perform its enzymatic function. So, to solve this problem, the Bacterius Thermus aquaticus was discovered through scientific research. It is a heat-resistant bacterium, and it has an enzymatic kit that allows it to live on the ocean floor up to 95 degrees. This bacterium has a metabolic system that contains a Thermophilus aquaticus polymerase, and thanks to this Taq polymerase, the bacterium Thermus aquaticus is able to function even in high temperature environments. The discovery allowed Kary Mullins, the person who uncovered the DNA PCR technique, to win the Nobel Prize for Chemistry in 1993.